Hello, my name is Ralph Isberg. I'm a professor of molecular biology and microbiology at Tufts University School of Medicine in Boston, and I'm an investigator of the Howard Hughes Medical Institute. And in this talk, which is the second talk of two in this series, I would like to discuss community behavior of an extracellular pathogen in tissues. In my first talk, I gave a very simple model for what differentiates a pathogen from a non-pathogen. In this talk, what you'll see is that the difference between a pathogen and non-pathogen are not so clear-cut. And what we'll see is that a pathogen can behave both like a non-pathogen as well as like a pathogen within host tissues. And so before I go into the details of this talk, I just want to give you the take-home messages, which I'll try to actually make you understand today. And the first thing that I'd like to discuss is that pathogen-specific proteins are expressed by only a fraction of bacteria. And the way you normally think of bacteria growing in culture is that a microorganism gives rise to two genetically identical microorganisms. And furthermore, these microorganisms then give rise to other organisms which have the same gene expression profiles as the initial parents. This is called a homogeneous population of bacteria growing in culture. And what I'd like to try to convince you of today is that when bacteria grow within tissue sites, that this probably is not the case. Instead, what happens is that bacteria may initially grow within a tissue and be homogeneous. They then give rise to occasionally bacterial cells which have a different transcriptional and translational profile than the parent. And then these organisms then go on to generate more of their kind as well as generate other organisms which may have other different transcriptional profiles. And therefore, what I'll try to convince you of is that bacteria growing within tissues are transcriptionally heterogeneous. The second thing that I'd like to convince you is that one of the reasons for this heterogeneity in gene expression is that the microenvironment in which these organisms grow controls the bacterial gene expression profile. And that can be seen in this slide. What I've described here for you are different microenvironments which the microorganism will encounter. When the organism grows and moves out of one microenvironment, it then encounters another microenvironment, and then that causes switching of the transcriptional profile so that it now assumes a transcriptional profile which is appropriate for that particular microenvironment. The bugs will continue to grow and expand, and then they'll encounter other microenvironments where they'll also similarly respond to that particular microenvironment. And then there's one more thing I'd like to point out, and that is that there's community behavior. So what is community behavior? And community behavior of bacteria is such that the bacteria will change the microenvironment in which other bacteria are able to grow. And this change can be due to a number of different things. One way in which this change can occur is that the organisms could secrete molecules which change the microenvironment, and other bacteria are able to respond to those molecules. But there's other ways in which this can occur, in which the microorganism is actually able to change the environment and deplete of certain molecules which are within the microenvironment, and then generate a whole new environment in which the molecular aspects of the environment are different than the original. As a result, then, the new bacteria which grow in this new environment have different transcriptional profiles which allow them to respond to this new microenvironment. All right, so the story I'd like to tell you is about an organism, Yersinia pseudotuberculosis. It's called an enteric organism because it causes disease through ingestion of contaminated foodstuffs. The organism is a gram-negative rod. That is, it's an organism which doesn't stain with gram stain. I've displayed this as red, which is typically what microbiologists display gram-negative rods. And then disease that's caused in the humans often results in growth in regional lymph nodes in the intestine. This is known as mesenteric adenitis. In the lab, we model this disease in mice. So with mice, what happens is we allow them to orally ingest the organism, or we will inject the organism intravenously. And then either route, the organism will enter into deep tissue sites and grow within the spleen. This particular disease then mimics what we call invasive diseases, in which organisms move from one tissue site to another. In this case, the tissue site which the organism is moving is from the intestine into deeper tissue sites, such as the spleen. We've been interested in this process for a number of years. And the way we look at this is that the organism is first ingested into the intestine. And what I've shown here is a diagram of what the small intestine looks like. The small intestine has crypts, which has its own specific crypt cells. It has microvilli, which are associated with the cells. Then it has the villus itself, which is up here at the top. In addition, there are regional lymph nodes. And the regional lymph nodes which we're most familiar with, which we work with, are regional lymph nodes called the Peyer's patches, as well as mesenteric lymph nodes. And the mesenteric lymph node is a small node which drains into the Peyer's patches. So both the intestinal epithelium, the intestinal lumen, and these regional lymph nodes are sites which can be exposed to the organism if the organism is able to move from the lumen of the intestine into these tissue sites. It's been known for a number of years, really since the 1950s, that when the organism is ingested by a host, the organism has the ability to move into both these tissue sites that are associated with the intestine, as well as deeper tissue sites such as the spleen. Now, we originally became interested in this a number of years when an MD fellow working in my lab, Penelope Barnes, who is currently an infectious disease specialist at the University of Oregon, was wondering whether when bacteria enter into these regional lymph nodes, whether they'll drain from the regional lymph nodes into deep tissue sites. And she got a very surprising result. And what she found is that the bacteria which grow within these regional lymph nodes are actually a different population of bacteria than the ones which are able to grow within deep tissue sites. So the way we look at this organism, the initial aspects of the disease are the following because of her studies. The bacteria enter into, are ingested by the host, in this case the mouse. Then the organism will then translocate across the epithelium. It translocates through a very special cell called the M cell, which are on the surface of Peyer's patches. And then we'll enter into Peyer's patch. And we believe that once the organism is found in the Peyer's patch, the immune response then will keep the replication in check. And even though you'll see very large boluses of growth of the organism in this site, the organism doesn't drain further from the site into deep tissue sites. It may drain from the Peyer's patches into the mesenteric lymph node, but there it's restricted and the immune response is very effective in keeping the organism from going any further into deep tissues. Instead, there's some kind of mysterious process that occurs, and we still don't know what it is, in which the organism is able to move across the intestinal epithelium, as can be seen over here, and then is able to find its way into the bloodstream. And then once in the bloodstream, the organism makes its way into deep tissue sites, such as the liver and spleen. So we look at this disease as a couple different things occurring simultaneously. An infection that occurs in regional lymph nodes, an infection that occurs in deep tissue sites. And there's actually little communication occurring between these two regions of the host. Now in the deep tissue site, this is a micrograph from my close colleague, Joan Mexis, who works at Tufts Medical School. This is a section through the spleen. On the top is a section in which an animal has been euthanized. The spleen has been frozen and frozen sections are taken. And then there is a histological stain which allows you to detect bacteria. And if you look in higher magnifications, you can see these foci of replication. In the dark dots are staining of replicating Yersinia pseudotuberculosis. And so we became very curious about the nature of these black spots. How do they go on to cause disease, and how do they spread throughout the spleen? So what does it mean to colonize a tissue? Now, the original view that we had from Penelope Barnes's work a number of years ago in my lab was that a few bacterial cells were seeding the spleen. And so typically she'd see between 1 and 4 cells initially seeing the spleen. And of course then the bacteria would go on to kill the host through unabated replication of the organism in this tissue site. So one way of looking at this is that these few organisms go on as if the spleen is simply a broth culture, and they continue to grow as if the spleen is a broth culture and then eventually overwhelm the spleen. One way of looking at this is it's very similar to inoculation of a broth culture in the lab. So this here is my colleague. He's taking a colony from a plate and then inoculating in a broth culture. This is similar to inoculation of the spleen that occurs after bacteria penetrate from the intestine and go into deep tissue sites. Then once the bacteria hit the spleen, in this case we've mimicked this by growing in broth culture, the bacteria will grow, and since the immune response isn't able to limit replication of the organism, it's a pathogen after all, the organism is able to grow in culture, and then we see a uniform broth culture within tissue sites. So that's one model, and I have to confess, this is the way I was thinking for a number of years. However, there's another model which maybe seemed more obvious to you, but wasn't obvious to me, and that is that the bacteria were initially seeding the spleen as between 1 and 4 cells, and then each of these cells were then giving rise to individual colonies. And so the disease was caused by colonization through a number of independent colonies forming in tissue sites. This could be mimicked also in the laboratory. So here you see my colleague then inoculating a petri dish. Again, this is similar to what we might see with seeding of the spleen. Bacteria are initially ingested by the host. Then the organism then is diluted and then goes into the spleen, and then individual cells seed the spleen. In this case, it would be the petri dish. Then once the petri dish is allowed to incubate for an appropriate amount of time, you get individual colonies coming up on the petri dish. So then the question is, are we getting a broth culture growing in the spleen, or are the bacteria growing as an agar plate? And so this is a very simple experiment, and the way we distinguish between these two models is the following. We take two bacterial strains that are genetically identical, except they differ in a single gene. One has a fluorescent protein called M. cherry, which glows red, as you might imagine. And the other one has a fluorescent protein that glows green, called GFP. We take these two bacterial cultures, and we mix them all up. So we have a culture that's full of red and green bacteria. Please excuse the use of red and green if you're colorblind. I'll try to talk this through for you. And then we take this mixed culture, and then we inoculate it intravenously into an animal, a few hundred or a few thousand bacteria into the animal. And then we allow some bacteria to seed the deep tissue site. And so in this, we've taken IV inoculation, and we use a mouse, and then we euthanize the mouse, and then we analyze the spleens by taking frozen sections of the spleen and then looking at these frozen sections in the fluorescence microscope. And you get a very obvious result, and that is it's the victory for the agar plate model. So as you can see right here, if we have a green colony, it's not mixed. If we have a red colony, it's not mixed. You might see some spread of the green cells into other tissue sites, but you don't see a mixture of red and green. So therefore, what happens when the bacteria are seeded into the spleen is as if they've been seeded onto an agar plate. Individual bacteria go on to form individual colonies, and there doesn't appear to be any mixing between the colonies. Once a colony is established, the bacteria do seem to be able to migrate from that colony into other sites and establish other colonies within the tissue. All right, now, when you look at this, you see there's a problem. And as I mentioned in the first talk, we distinguish pathogens from non-pathogens by the virulence proteins which pathogens make. And what determines whether a pathogen will make a virulence protein is whether it's actually interacting directly with host tissues. But what you can see here is when a colony forms, not all the bacteria are interacting with host cells. We have bacteria internal to these small colonies, and all they're seeing is other bacteria in the colony. Whereas we have other bacteria that are at the peripheries of this colony, and they're clearly interacting with host cells. And the host cells that we see that surround these bacteria, and this will be demonstrated later in this talk, these host cells are neutrophils, phagocytic cells, which are trying to destroy the microorganism. And the neutrophils are displayed here in blue. They're displayed here in blue because we've stained their nuclei with a DNA stain. All right, so now the question is, what is it about these bacteria in these different environments? Do they behave differently? So when we look at this, there's clearly something going on. The first thing that you notice in this particular tissue site is that we've recruited neutrophils to this site, and the bacteria are still alive. And also what you see is that the bacteria are encountering the phagocytic cell, and there are a subset of bacteria which are interacting with the phagocytic cell, but the bacteria are not being phagocytized, they're simply bound to the surface. And the second thing you notice is that there are bacteria within this colony which are not interacting with the phagocytic cell at all. So the question is, why do you have one set of bacteria directly interacting with host cells and the other one that seems to be naive to these cells? So clearly, this pathogen is doing something to the phagocyte. It's preventing phagocytosis. And the way it does that is it has a type 3 secretion system. A type 3 secretion system is one of the specialized secretion systems which I described in the previous talk. And the type 3 secretion system allows a movement of a protein into the host cell, which interferes with phagocytosis. And the way these proteins are able to move from the bacterium into the host cell is through this needle-like structure which is on the surface of the bacterium, in which bacterial proteins which interfere with phagocytosis of the host cell move through this structure across the plasma membrane of the host cell and then are able to get a cytoplasmic target a cytoplasmic protein within the host phagocyte. This is the structure of these particular secretion complexes was initially described in Jorge Galan's lab, and there's some beautiful structures that have been now demonstrated in the lab of Marlovitz and co-workers. And this is an EM structure of such a complex. As you can see, there's a base which is through the envelope of the bacterial cell, and then there's this needle which sticks out from the surface of the bacterial cell into the host cell. All right, so now the secreted protein goes through the type 3 translocation apparatus, and what's probably the most important protein for this process is the protein YAP-E, which is one of these proteins that targets small GTPases in the host cell and inactivates Rho family members to prevent phagocytosis. So what you see here is YAP-E is being secreted by the bacterium. It's now interfering with phagocytosis by the neutrophils that are surrounding the host cell, and then you see this frustrated phagocytosis event. On the other hand, the bacteria which are within the colony do not have to deal directly with the phagocyte, and therefore they may not need the transcriptional program which makes these proteins which have to inactivate the neutrophil. So what we wanted to ask then is there spatial regulation? So do the bacteria which interact directly with the neutrophil have a different transcriptional profile than the bacteria which are growing within the center of these host cells? And so we believe that that's probably the case, and we had reason to believe this was the case based on previous work from workers studying organisms growing within the lumen of the intestine. And in this particular work, which was done by Sansonetti and co-workers, Christoph Tang, what they did is they fed another pathogen, Shigella flexneri, into animals. And they noticed that bacteria that were growing near the interface of intestinal epithelium and in the lumen seemed to be seeing a different environment in regards to oxygen tension. And the way this could be seen is with one of these fluorescent proteins, GFP, which only folds if you have sufficient oxygen in the environment. And what they found is that in the case of these GFP encoding Shigella, bacteria which were growing within the lumen did not fluoresce. But as you got closer and closer to the lumen of the intestine, what happens is they begin to fluoresce. And the reason why is because the oxygen tension here near the intestinal epithelial cells is much higher than in the lumen of the intestine, which is basically anaerobic, and the GFP will not fold. And so this colored our way of looking at things. And so we asked if a similar thing could be going on, but it's a little different than the oxygen environment. What we're seeing here is we're seeing a cell interactive environment as well as an environment in which the bacteria are growing within the center. So now the second thing that we are interested in then is whether there's community behavior. And we define community behavior in the following. And that is that the activity of one population causes a second population to be differentially regulated. So as you recall from the early slides, bacteria which would be growing associated with tissues, in this case neutrophils, might change the environment so that the bacteria growing within the center of the colony then are experiencing a different environment. And as a result of this different environment then, they undergo a different transcriptional profile that looks very different from the bacteria which are growing associated with host cells. Okay, so let's go and talk about the first point. And again, YAP will interact with the phagocyte, interfere with phagocytosis. And so one of the interesting aspects of the regulation of the gene that encodes this protein is that it was been shown by Hans Wolfwatz and co-workers almost 20 years ago that the gene for this protein is upregulated, so the gene is transcribed at higher levels when bacteria are growing in contact with host cells than when they're not. In this simple experiment from Wolfwatz's lab, he takes bacteria in which a luciferase reporter is fused to the gene for YAP, and then incubates it with cultured cells, in this case it's HeLa cells. And then the bacteria which are associated simply with the extracellular matrix are not turning on this gene, but those which are associated with the cells are. So then what we asked is this really what's going on in tissues? And so we made a similar type of reporter, and this reporter actually has two different fluorescent proteins in it. One fluorescent protein has GFP, and GFP is just made constitutively, so it's always on. And the other has cherry. Cherry is a red fluorescent protein, and it's under the transcriptional control of the YAP gene. Otherwise, this organism is totally wild type. It has all the components necessary for causing disease. Then we take this particular construct. We then inject animals intravenously. We allow infection to go on for 3 days. We then harvest the spleens, and we perform histology on frozen sections using fluorescent microscopy. Usually, we use confocal microscopy to do this. And you see a very striking result. Again, red is the YAP gene. Green is just constitutive expression. And what you can see is that on the edges of these colonies, the bacteria glowing red, they're in contact with the host cells, with the neutrophils, they're turned on. But the bacteria in the center have a different transcriptional profile. They're green. They're not turning on this gene. So they're in a protective environment in which they're not directly interacting with phagocytic cells. They're growing, in essence, in some ways, very similarly to bacteria which are not growing in tissues. So there's the contact with the host cells, and those are the bacteria that are internal. Now, this gives us two transcriptionally distinct populations. And it's clear that it looks like there's an altruistic effect, so that the bacteria directly interacting with the host cells are changing the environment so that the bacteria in the center are able to actually avoid phagocytosis. And not only does avoid phagocytosis means that they don't have to worry about being killed by the phagocyte, it means they can undergo a transcriptional profile, which starts to look a little bit like what a non-pathogen looks like, in that they may not be making some of the genes that are expressing some of the genes that are important for disease. But the question is, there's other things going on in tissues. And I mentioned in the previous talk when these foci start to get larger, what happens is there's secondary responses in which the host tries to clear out microorganisms. One of the secondary responses is nitric oxide. So there's cells that might come in and make soluble mediators to actually start to clear of the organism. And so what we wanted to ask is this more heterogeneous than we even thought initially? Is there actually more than one population? So nitric oxide, as I mentioned previously, would be a very important antimicrobial molecule that these cells might see. It can diffuse into the center of these microcolonies, and so the bacteria don't actually have to directly interact with phagocytes in order to see this antimicrobial molecule. And nitric oxide, of course, is made by inducible nitric oxide synthase. So now we believe that Yersinia has to have strategies for dealing with nitric oxide. And the reason for believing this is that the very closely related, shockingly closely related organism, Yersinia pestis, which causes bubonic plague, which is a very disease that's very different from Yersinia pseudotuberculosis, appears to require strategies for dealing with the nitric oxide response. And in enteric organisms, that is gram-negative organisms which are similar to E. coli, the most important way in which these organisms are able to inactivate nitric oxide is through the use of this protein called HMP, which is a flavoprotein which inactivates nitric oxide. And what Saban and co-workers showed a number of years ago is that if you don't have HMP, then Yersinia pestis, which causes a lethal disease in mice, shows delayed or no death in other animals if the bacteria are unable to make the HMP protein. So we decided to investigate this in detail, and there was reasons for thinking that HMP would be an important protein to look at, because HMP is a gene that's highly regulated by the presence of nitric oxide. So in bacteria, nitric oxide is able to regulate a number of genes. The HMP protein, which takes nitric oxide and oxygen and inactivates it by making NO3-, is a flavoprotein, as described right here. And it's regulated by a transcriptional regulator called NSRR, which recognizes high levels of nitric oxide in tissues. And then there's a conformational change in NSRR, which activates the gene for HMP and it then gets turned on. That means we can use the gene for HMP as a reporter for nitric oxide concentration in host tissues. So the reporter we made was very similar to the one we showed before, and that is that we have a constitutively expressed GFP, and then we have cherry under the control of the HMP gene. So what we see is something very similar to what we saw with YAP-E, is that there is an extreme gradient of expression of the HMP gene. On the edges, what we see, and bacteria, they're migrating into tissues. The bacteria are all red, so they have high HMP expression. However, in the center, you see the beginning of a gradient. You see yellow, which means that the red is expressed at lower levels in these cells. And then in the center, frankly, you see green, which means we see the green fluorescent protein, but we don't see the red fluorescent protein in the center of these colonies. Clearly, as the bugs migrate into tissues, they're able to see nitric oxide. This can also be displayed another way, where we ratio the cherry to the GFP in this ratiometric display. Red means that you have very high cherry levels. Blue means that the levels of GFP relative to cherry are very high. And this is over a 10-fold gradient based on this ratiometric assay, in which we can see that there's very low expressions of HMP in the center of the colony and very high on the edges. This can also be displayed in another way, in which we do 3D imaging of this particular structure. Okay, so now the question is, why is there this steep gradient? And there's two explanations for why the gradient could be steep. One reason is that there's this gradient of expression where the bugs go from red to yellow to green because the NO gradient in the host tissues is very steep. As a result, the bacteria growing within the center of the colony don't see NO. But what we were more curious about is whether we could find an example of community behavior. That is, are the bacteria on the edges facilitating growth of bacteria in the center because they're removing nitric oxide from the center of the colony? That's displayed here. Bacteria growing near a neutrophil make the HMP protein. They then remove or reduce the amount of NO in the environment. Blue is high NO, white or light blue is low NO. And then as the NO gets lower, then there's no NO in the system where the bacteria are growing, and then you no longer see HMP cherry on anymore, and the bacteria are growing relatively free of NO because of this community behavior. So how do we actually test this, distinguish between these two models? So the way we distinguish between these two models, we take advantage of the fact that HMP actually inactivates NO. So in a wild-type organism, the HMP will take NO and reduce the amount of NO in the environment. And in the wild type, what will happen if there's a steep NO gradient? We see a steep NO gradient with the wild type because the gradient is simply steep. And if you have an HMP mutant, it doesn't matter whether it's inactivating the NO or not. There's simply a steep gradient from the presence of neutrophils making it high near them, and then there's loss of NO deeper into the colony. On the other hand, if there's community behavior, you'll see a different result. In the wild type, what happens is the HMP protein would be removing the NO from the environment and then allowing bugs to grow in the absence of HMP expression. And then in the case of the mutant, the HMP is no longer removing NO from the tissues. And now what happens is what we see is that the bacteria now are able to induce the HMP expression because NO is not removed. All right, so this is a very simple experiment, and we get a relatively simple answer. And the answer is the following, that the gradient that we see is now gone. So now in this experiment, what we do is we've inverted the GFP, and what we've done here is we've taken the HMP minus and the HMP plus. And what you see is the HMP plus, you see cherry expression on the edges. And then in the center, there's little expression at all. So we've seen this before. Now what happens in the case of, if we have a HMP mutant, this gradient is gone, and we start now to see cherry now expressed at high levels in the center of these colonies. This can be seen in this 3D reconstruction. So right here, the way we've displayed this data is if you have high fluorescence, in the x-axis you see high peaks, and if you have low levels of expression, you see absence of peak. What you can see is in the wild-type situation, there's a loss of expression in the center, but in the deletion, where we're allowing NO to get in the center of the colony, we're seeing expression throughout the colony. So we view this as community behavior. All right, now, the question is, why is this? So one of the most interesting aspects of this is the fact that the bacteria on the edges of the colony are facilitating the bacteria in the center to grow. So that you actually need that expression on the outside in order to cause disease. And that can be seen here. I showed you pictures of animals in which we sectioned through the spleen 3 days after infection. And the size of the colonies in either the wild type or the delta HMP mutant look almost identical. However, if you wait 5 days after infection, what happens is the colonies continue to grow in the wild type situation, but in the mutant, the colonies start to break apart. What we find interesting about this is that the ability to establish this colony and maintain the colony is dependent on only a subset of bacteria expressing this gene, because these bacteria in the center aren't expressing it. So if the bugs on the outside are unable to express this particular protein, then what happens is the whole colony breaks apart. All right, now, so the question is, are we actually seeing two signals? Is there a cell-directed, a signal that's coming direct from the cell? Or is there actually a soluble signal and a signal that's coming from the host cell? And so we look at that as either cell contact and NO as a single signal that might be coming in the bugs on the edges responding to, whereas the bugs in the center are responding to no signal at all, and they're growing something in between a pathogen and non-pathogen. Now the alternative is that there's actually three signals going on, and that there's a cell contact and an NO signal going on in the edges because these cells are seeing NO as well as cell contact. But then there's other bugs which are within this particular tissue site, which are seeing NO, but they're not seeing the host cell. And then perhaps there's a third population which is seeing neither the host cell nor NO. And so then we draw this, therefore, as three populations in which there's bacteria on the edges directly contacting the host. And then there's other bacteria which are not directly contacting the host cell, but they might be seeing NO. And then ones in the center, they're seeing neither NO nor the host cells. So now to distinguish between these two models, we set up another reporter situation. So in this reporter situation, we have the cell contact signal reporter, YAP-E, now in red, and GFP now fused to HMP. We put them both into either a wild type or a delta HMP strain, and then we ask whether we see a gradient of expression like we'd seen before. If these two proteins, these two genes are now responding to the identical signals, the gradient that we see that gets removed when we're studying the HMP gene should also be removed when we look at expression of YAP-E. However, that's not what we see. What you see is that now the green GFP is HMP. What you see is that there's HMP expression throughout the colony, but YAP-E is still expressed at very high levels around the edges where it's in contact with host cells. So these two proteins are responding, therefore, to different signals. Now in the wild type, again, you see something similar to what we'd seen before. You see this extreme gradient of expression of HMP. All right, so now we believe that we've got three different signals. There's ones that are missing both the cell contact signal and the NO signal. It doesn't mean they're not seeing some other signals we haven't identified. And in fact, my lab's trying to identify some other signals that these ones in the center might be saying that might distinguish these cells from organisms that are growing simply in culture. There's ones that are responding to an NO signal. They're not in contact with a host cell, but they're turning on their high GFP levels. And then the ones that are high GFP levels are on the edges are responding to both the cell contact signal and to an NO signal. So now the question is, where is the nitric oxide coming from? So it could be coming directly from the host cells which the organism is interacting with, or it could be coming from somewhere else. And the most straightforward way to find out where the NO is coming from is to study inducible nitric oxide synthase, the protein which makes nitric oxide. And so we can determine where the nitric oxide is coming from by simply probing with antibody directed against inducible nitric oxide synthase and identifying cells which are expressing this protein in tissues. And so what I'm going to show you is a series of panels in which I show you a typical colony of Yersinia pseudotuberculosis growing in tissues. In blue are the bacteria, which we stain with antibody directed against the bacteria, and you see this nice colony. And then the second thing I'm going to show you is a stain now for inducible nitric oxide synthase. And surprisingly, what you see is that although bacteria are growing within this colony here, there's a gap. And as I'll show you on the next slide, this gap actually has cells. But this gap where the cells are not making inducible nitric oxide synthase. Instead, the inducible nitric oxide synthase are coming from cells in the periphery. These cells out here that are making inducible nitric oxide synthase, we believe, are macrophages and lymphocytes. Now, if we stain with neutrophils, which I've incessantly told you the bugs are interacting with without actually giving you evidence for, what we find is that the neutrophils are directly interacting with the host, with the bacteria, but the neutrophils that are interacting with the bacteria are indeed not making nitric oxide synthase. So the nitric oxide synthase is coming from afar and then managing to migrate through the neutrophils and actually attacking the bacteria on the edges. Okay, so this sort of summarizes what I'd like to communicate with you today, and that is that the nitric oxide producing cells are very far away, and the nitric oxide producing cells are not directly in contact with the host cells, but the bacteria is still responding to the nitric oxide. When they respond to nitric oxide, the nitric oxide gradient, which is displayed here as blue high to yellow low, when the bacteria encounter it, the HMP protein inactivates it, allowing bacteria in the center of the colony to be naive for nitric oxide and not see it. Furthermore, the bacteria in the center are also protected from phagocytes because the ones on the edges are acting to interfere with phagocytosis and prevent their access to the bacteria in the center of the colony. All right, so now, why do we think this is important? Do we think that this is a general model for how all pathogens work? And so our way of thinking about this is colored by work that was published last year in Nature by Hart and Ackerman and co-workers, who demonstrated that in the case of salmonella, that if bacteria which are inoculated into an animal, in this case a mouse, and in this case it's salmonella, bacteria will make a virulence-dependent cascade, which will allow them to invade into host tissues and bypass the immune response. What they demonstrated is that all the bacteria in the population actually express genes associated with virulence. What they saw is that if you model this, that the number of bacteria that are fully virulent will decrease over time, and instead they'll be replaced by avirulent derivatives of the organism. So by having a virulence cascade, which is on in all the organisms in a tissue, you actually select now for faster growing non-pathogenic variants, which will overgrow the population. On the other hand, if you build a pathogen a little more wisely, so that a proportion of the organisms are not producing virulence-associated proteins, then you see a higher persistence of the fully virulent organism. So what we believe is that the reason why pathogens have this heterogeneous population is that it's able to now allow expression of proteins on the edges which result in the organisms actually growing slower but dealing directly with the host immune function. And then there's another population protected. And this faster growing population, which doesn't have to directly interact with host cells, can outcompete avirulent derivatives because they're able to grow just as fast as some avirulent derivative. So the fitness cost that organism might have from making virulence-associated proteins is partially overcome from the ability of these organisms to grow heterogeneously in tissues. So let me summarize now with the 3 points that I want to make, is that pathogen-specific proteins are only expressed by a fraction of bacteria, and we believe the reason for that is because this allows them to have a fitness cost which is less than they would have than if all the bacteria in the population actually expressed virulence-associated proteins. The environment in which the bacteria encounter is different in different sites within the tissue, and that includes in the narrow environment in which the organism is growing. And then finally, as the bugs move into other sites, there are different environments which the microorganism encounters. And when it encounters these different sites, it will now respond with a different transcriptional profile. So finally, I'd like to thank the people who worked on this. Sino Mohammadi was a graduate student and a postdoc in my lab who did the initial reporters to allow following of gene expression in tissues. And of course, I'd like to thank Penelope Barnes, who was an MD fellow in my lab, who got us thinking about colonies within tissues. And Molly Bergman and Greg Crimmins did the initial studies which led to most of this work which I've described, which was performed by Kim Davis in my lab. I've acknowledged her work throughout on the slides with her name there. And in addition, I'd like to thank Carrie Sheehan, who's developed analysis of type 3 translocation in my lab, as well as special thanks to Joan Mexis, who is far more expert at animal infection models than I am, and much of what I've learned has been learned from her. And then my funding is from NIAID, which has funded my lab on Yersinia for over 20 years, as well as Howard Hughes Medical Institute. Thank you.